Chapter 5 Morphology of Flowering Plant. The wide range in the structure of higher plant will never fail to fascinate us, even though the angiosperm shows such a large diversity in external structure or morphology. They are all characterized by presence of root, stems, leaves, flowers, and fruits. In chapter 2 and 3, we talked about classification of plant based on morphological and other characteristics. For any successful attempt at classification and at understanding any higher plant, or for that matter any living organism, we need to know, understand technical terms and standard definition. We also need to know about the possible variation in different parts found as adaptation of the plant to their environment. Example, adaptation to various habitat for protection, climbing, storage, etc. If you pull out any weed, you will see that all of them have root, stems, and leaves. They may be bearing flower and fruit. The underground part of the flowering plant is the root system, while the portion above the ground forms the shoot system. Figure 5.1. The root. In majority of the dicotyledonous plants, the direct elongation of the radicle leads to the formation of primary root, which grow inside the soil. It bear lateral root of several order that are referred to as secondary, tertiary, etc. roots. The primary roots and its branches constitute the taproot system as seen in the mustard plant. Figure 5.2a. In monocotyledonous plants, the primary root is short-lived and is replaced by a large number of roots. This root originate from the base of the stem and constitute the fibrous root system as seen in the wheat plant. Figure 5.2b. In some plant like grass, monstera, and the banyan tree, root arise from parts of the plant other than the radicle and are called adventitious root. Figure 5.2c. The main function of the root system are absorption of water and mineral from the soil, providing a proper anchorage to the plant part, storing reserve food material, and synthesis of plant growth regulators. Region of the root. The root is covered at the apex by a thimble-like structure called the root cap. Figure 5.3. It protects the tender apex of the root as it makes its way through the soil. A few millimeters above the root cap is the region of meristematic activity. The cells of this region are very small, thin-walled, and with dense protoplasm. They divided rapidly. The cells proximal to this region undergo rapid elongation and enlargement and are responsible for the growth of the root in length. The region is called the region of elongation. The cell of the elongation zone gradually differentiate and mature. Hence, the zone proximal to region of elongation is called the region of maturation. From this region, some of the epidermal cells form very fine and delicate thread-like structure called root hair. The root hair absorb water and mineral from the soil. The stem. What are the features that distinguish a stem from a root? The stem is the ascending part of the axis bearing branches, leaves, flowers, and fruits. It develops from the plumule of the embryo of a germinating seed. The stem bear node and internode. The region of the stem where leaves are borne are called nodes, while internodes are the portion between two nodes. The stem bear buds which may be terminal or axillary. Stem is generally green when young and later often become woody and dark brown. The main function of the stem is spreading out branches bearing leaves, flowers, fruits. It conducts water, minerals, and photosynthesis. Some systems perform the function of storage of food, support, protection, and of vegetative propagation. The leaf. The leaf is a lateral, generally flattened structure borne on the stem. It develops at the node and bear a bud in its axil. The axillary bud later develop into a branch. Leaves originate from shoot apical meristem and are arranged in an acropetal order. They are the most important vegetative organ for photosynthesis. A typical leaf consists of three main parts: leaf base, petiole, and lamina. Figure 5.4a. The leaf is attached to the stem by the leaf base and may bear two lateral small leaf-like structure called stipules. In monocotyledonous, the leaf base expands into a sheath covering the stem partially or wholly. In some leguminous plants, the leaf base may become swollen, which is called the pulvinus. The petiole help hold the blade to light. Long, thin, flexible petiole allow leaf blade to flutter in wind, thereby cooling the leaf and bringing fresh air to leaf surface. The lamina or the leaf blade is the green expanded part of the leaf with veins and veinlets. There is usually a middle prominent vein which is known as the midrib. Veins provide rigidity to the leaf blade and act as channel of transport for water, minerals, and food materials. The shape, margin, apex, surface, and extent of incision of lamina varies in different leaves. Venation. The arrangement of veins and the venlets in the lamina of leaf is termed as venation. When the venlet form a network, the venation is termed as reticulate. Figure 5.4b. When the veins run parallel to each other within a lamina, the venation is termed as parallel. Figure 5.4c. Leaves of dicotyledonous plant generally possess reticulate venation, while parallel venation is the characteristic of most monocotyledonous type of leaves. A leaf is said to be simple when its lamina is entire or when incised. The incision do not touch the midrib. When the incision of the lamina reach up to the midrib, breaking it into a number of leaflets, the leaf is called a compound. A bud is present in the axial of petiole in both simple and compound leaves, but not in the axial of leaflet of the compound leaf. The compound leaf may be of two types. Figure 5.5. In a pinnately compound leaf, a number of leaflets are present on a common axis, the rachis which represent the midrib of the leaf as in neem. In palmately compound leaves, the leaflets are attached at a common point, example, at the tip of petiole as seen in silk cotton. Phyllotaxy. Phyllotaxy is the pattern of arrangement of leaves on the stem or branch. This is usually of three types: alternate, opposite, and roll. Figure 5.6. In alternate type of phyllotaxy, a single leaf arises at each node in alternate manner as in China rose, mustard, and sunflower plant. In opposite type, a pair of leaves arise at each node and lie opposite to each other, as in calotropis and guava plant. If more than two leaves arise at a node and form a roll, it is called roll, as in Alstonia. The inflorescence. A flower is a modified shoot wherein the shoot apical meristem changes to floral meristem. Internode do not elongate and the axis get condensed. The apex produce different kind of floral appendages laterally at successive node instead of leaves. When a shoot tip transform into flower, it is always solitary. The arrangement of flowers on the floral axis is termed as inflorescence, depending on whether the apex get developed into a flower or continue to grow. Two major type of inflorescence are defined: racemose and cymose. In a racemose type of inflorescence, the main axis continue to grow. The flower are borne laterally in an acropetal succession. Figure 5.7. In a cymose type of inflorescence, the main axis terminate in a flower, hence is limited in growth. The flower are borne in a basipetal order. Figure 5.7. The flower. The flower is the reproductive unit in the angiosperm. It is meant for sexual reproduction. A typical flower has four different kinds of rolls arranged successively on the swollen end of the stalk or pedicle, called thalamus or receptacle. These are calyx, corolla, endoecium, and gynoecium. Calyx and corolla are accessory organ, while endoecium and gynoecium are reproductive organ. In some flower like lily, the calyx and corolla are not distinct and are termed as prianth. When a flower has both endoecium and gynoecium, it is bisexual. A flower having either only stamen or only carpel is unisexual. In symmetry, the flower may be actinomorphic radial symmetry or zygomorphic bilateral symmetry. When a flower can be divided into two equal radial halves in any radial plane passing through the center, is said to be actinomorphic. Example: mustard, datura, chili. When it can be divided into two similar halves only in one particular vertical plane, it is a zygomorphic. Example: pea, gulmohar, bean, cassia. A flower is asymmetric irregular if it cannot be divided into two similar halves by any vertical plane passing through the center, as in canna. A flower may be trimerous, tetramerous, or pentamerous when the floral appendages are in multiple of 3, 4, or 5, respectively. Flowers with bracket reduced leaf found at the base of the pedicle are called bracteate, and those without bracket, ebracteate. Based on the position of calyx, corolla, and endoecium in respect of the ovary on thalamus, the flowers are described as hypogynous, perigynous, and epigynous. Figure 5.9. In the hypogynous flower, the gynoecium occupy the highest position while the other part are situated below it. The ovary in such flower is said to be superior. Example: mustard, China rose, and brinjal. If gynoecium is situated in the center and other part of the flower are located on the rim of the thalamus almost at the same level, it is called perigynous. The ovary here is said to be half inferior. Example: plum, rose, peach. In epigynous flower, the margin of the thalamus grow upward, enclosing the ovary completely and getting fused within it. The other part of flower arise above the ovary, hence the ovary is said to be inferior, as in flower of guava and cucumber and the ray florets of sunflower. Parts of flower. Each flower normally has four floral rolls. Calyx, chlora, endoecium, and gynoecium. Figure 5.10. Calyx. The calyx is the outermost roll of the flower, and the members are called sepals. Generally, sepals are green, leaf-like, and protect the flower in the bud stage. The calyx may be gamisophalous, sepals united, or polysepalous, sepals free. Chlorella. Chlorella is composed of petals. Petals are usually brightly colored to attract insects for pollination. Like calyx, chlorella may also be gamopetalous, petalous united, or polypetalous, petalous free. The shape and color of chlorella vary greatly in plant. Chlorella may be tubular, bell-shaped, funnel-shaped, or wheel-shaped. Estivation, the mode of arrangement of sepal or petal in floral bud with respect to the other member of the same role is known as estivation. The main type of estivation are volvate, twisted, imbricate, and waxillary. Figure 5.11. When sepal or petals in a roll just touch one another at the margin without overlapping, as in calotropis, it is said to be volvate. If one margin of the appendages overlap that of the next one, and so on, as in China rose, ladyfinger, and cotton, it is called twisted. If the margin of sepal or petal overlap one another but not in any particular direction, as in cassia and gulmohar, the estivation is called imbricate. In pea and bean flower, there are 5 petals. The largest standard overlaps the two lateral petal wings, which in turn overlap the two smallest anterior parts keel. This type of estivation is known as waxillary or papillonoecious. Androecium. Androecium is composed of a stamen. Each stamen, which represents the male reproductive organ, consists of a stalk or a filament and an anther. Each anther is usually bilobed, and each lobe has two chambers. The pollen sac, the pollen grain are produced in pollen sac. A sterile stamen is called staminode. Stamen of flower may be united with other members such as petal or among themselves. When stamens are attached to the petal, they are epipetalous as in brinjal, or epiphyllous when attached to the perianth as in the flower of lily. The stamen in a flower may either remain free, polyndrous, or may be united in varying degrees. The stamen may be united into one bunch or one bundle, monodulphus as in China rose, or two bundle, diadelphus as in pea, or into more than two bundle, polydulphus as in citrus. There may be a variation in the length of filament within a flower as in salvia and mustard. Gynoecium. Gynoecium is the female reproductive part of the flower and is made up of one or more carpel. A carpel consists of three parts, namely stigma, style, and ovary. Ovary is the enlarged basal part on which lie the elongated tube, the style. The style connects the ovary to the stigma. The stigma usually at the tip of the style and is the receptive surface for pollen grain. Each ovary bear one or more ovule attached to a flattened cushion-like placenta. When more then one carpel is present, they may be free as in lotus and rose and are called apocarpus. They are termed syncarpus when carpel are fused as in mustard and tomato. After fertilization, the ovule develop into seed and the ovary mature into fruit. Placentation. The arrangement of ovule within the ovary is known as placentation. The placentation are of different type, namely marginal, axile, parietal, basal, central, and free central. Figure 5.12. In marginal placentation, the placenta forms a ridge along the ventral suture of the ovary, and the ovule are borne on this ridge, forming two rows as in P. When the placenta is axile and the ovule are attached to it in a multilocular ovary, the placentation is said to be axile, as in China rose, tomato, and lemon. In parietal placentation, the ovule develop on the inner wall of the ovary or on peripheral part. Ovary is one chambered, but it becomes two chambered due to the formation of the false septum. Example: mustard and argemone. When the ovule are borne on central axis and septa are absent, as in dianthus and primrose, the placentation is called free central. In basal placentation, the placenta develops at the base of ovary and a single ovule is attached to it, as in sunflower, marigold. The fruit. The fruit is a characteristic feature of the flowering plant. It is a mature or ripened ovary developed after fertilization. If a fruit is formed without fertilization of the ovary, it is called a parentocarpic fruit. Generally, the fruit consists of a ball or pericarp and seeds. The pericarp may be dry or fleshy. When epicarp is thick and fleshy, it is differentiated into the outer epicarp, the middle mesocarp, and the inner endocarp. In mango and coconut, the fruit is known as drupe. Figure 5.13. They develop from monocarpellary superior ovaries and are one-seeded. In mango, the pericarp is well differentiated into an outer thin epicarp, a middle fleshy edible mesocarp, and an inner stony hard endocarp. In coconut, which is also a drupe, the mesocarp is fibrous. The seed The ovule after fertilization develop into seeds. A seed is made up of a seed coat and embryo. The embryo is made up of a radicle and embryonal axis and one, as in wheat, maize, or two cotyledons, as in gram and pea. The structure of a dicotyledonous seed. The outermost covering of a seed is the seed coat. The seed coat has a two layer, the outer testa and the inner tegmen. The hilum is a scar on the seed coat through which the developing seed were attached to the fruit. Above the hilum is a small pore called the microphyll. Within the seed coat is the embryo, consisting of an embryonal axis and two cotyledons. The cotyledons are often fleshy and full of reserve food material. At the two ends of the embryonal axis are present the radicle and the plumule. Figure 5.14. In some seed, such as castor, the endosperm, formed as a result of double fertilization, is a food storing tissue and called endospermic seed. In plants such as bean, gram, and pea, the endosperm is not present in mature seed and such seed are called non-endospermous. Structure of monocotyledonous seed. Generally, monocotyledonous seed are endospermic, but some, as in orchids, are non-endospermic. In the seed of cereals such as maize, the seed coat is membranous and generally fused within the fruit wall. The endosperm is bulky and store food. The outer covering of endosperm separates the embryo by a proteinaceous layer called aleurone layer. The embryo is small and situated in a groove at one end of the endosperm. It consists of one large and shield shape known as scutellum and a short axis with a plumule and a radicle. The plumule and radicle are enclosed in sheath which are called caloptile and chlorohizia respectively. Figure 5.15. Semi-technical description of a typical flowering plant. Various morphological features are used to describe a flowering plant. The description has to be brief in a simple and scientific language and presented in a proper sequence. The plant is described beginning with its habit, vegetative character, root, stem and leaves, and then floral character, inflorescence and flower parts. After describing various parts of a plant, a floral diagram and a floral formula are presented. The floral formula is represented by some symbol. In the floral formula, BR stand for bracteate, K stand for calyx, C for corolla, P for perianth, A for androecium, and G for gynoecium. G for superior ovary and G inferior ovary, male for female for bisexual plant for actinomorphic, and percentage for zygomorphic nature of flower. Fusion is indicated by enclosing the figure within bracket and adhesion by a line draw above the symbol of the floral part. A floral diagram provide information about the number of parts of a flower, their arrangement and the relation they have one another. Figure 5.16. The position of the mother axis with respect to the flower is represented by a dot on the top of the floral diagram. Calyx, corolla, androecium and gynoecium are drawn in successive row, calyx being the outermost and the gynoecium being in the center. Floral formula also show cohesion and adhesion within parts of rows and between rows. The floral diagram and floral formula in figure 5.16 represents the mustard plant family Brassicaceae Solanaceae. It is a large family commonly called as the potato family. It is widely distributed in tropic, subtropic and even temperate zone. Figure 5.17. Vegetative character. Plant mostly herbs, shrubs and rarely small trees. Stem herbaceous, rarely woody, aerial, erect, cylindrical, branch, solid or hollow, hairy or glabrous underground stem. In potato, solanum tubersome. Leaves alternate, simple, rarely pinnately compound, stipulate, venation, reticulate. Floral characters: inflorescence solitary, axillary, or cymose, as in solanum. Flower bisexual, actinomorphic. Calyx sepal 5 united, peristent, valvate, aestivation. Corolla petals 5 united, valvate, aestivation. Androecium stamen 5 epipetalous. Gynoecium bicarpellary, obligate placed. Syncarpus ovary superior, bilocular. Placenta swollen with many ovule, axile. Fruit berry or capsule. Seed many, endospermous. Floral formula. Economic importance: many plants belong to this family are source of food: tomato, brinjal, potato, spice, chili, medicine, belladonna, ashwagandha, fermentary, tobacco, ornamental, petunia. Summary: flowering plants exhibit enormous variation in shape, size, structure, mode of nutrition, lifespan, habit, and habitat. They have well-developed root and shoot systems. Root system is either taproot or fibrous. Generally, dicotyledonous plants have taproot, while monocotyledonous plants have fibrous root. The root in some plants get modified for storage of food, mechanical support, and respiration. The shoot system is differentiated into stem, leaves, flowers, and fruit. The morphological feature of stems like the presence of node and internode, multicellular hair, and positively phototropic nature help to differentiate the stem from root. Leaf is a lateral outgrowth of stem developed exogenously at the node. These are green in color to perform the function of photosynthesis. Leaves exhibit marked variation in their shape, size, margin, apex, and extent of incision of leaf blade lamina. The flower is a modified shoot meant for sexual reproduction. The flowers are arranged in different type of inflorescences. They exhibit enormous variation in structure, symmetry, position of ovary in relation to other part, arrangement of petal, sepal, ovule, etc. After fertilization, the ovary is modified into fruit and ovules into seed. Seed either may be monocotyledonous or dicotyledonous. They vary in shape, size, and period of viability. The floral characteristic from the basis of classification and identification of flowering plant. This can be illustrated through semi-technical description of families. Hence, a flowering plant is described in a definite sequence by using scientific term. The floral features are represented in the summarized form as floral diagram and floral formula.